Well, thank you everyone for joining today. I am so excited for this webinar. I love that we're spotlighting research on breast cancer during the month of October. To introduce who I am, I'm Molly Clemens. I work for Altium. We're the distributors for cutting-edge disruptive biotechnologies, and we've been a long-time partner with Phase Holographic Imaging. So Phase Holographic Imaging, or PHI, they manufacture the HoloMonitor. Back in March of 2025, I had the pleasure of meeting the Luna Lopez lab for the first time over at California State University, San Marcos. As soon as the system was installed, they were off and running. And within a few months, I was back on campus for the CS USM Summer Scholar Showcase. This is where I first met Faheema and saw her poster. So to give you some background on Faheema Sahib, she's a biotechnology student focusing her research on cancer biology. She was previously a Bears esteemed NIH scholar, so she was not only conducting research, she was also mentoring students. She's currently working as a research assistant in the Luna Lopez lab, where she focuses on using holographic microscopy and computational analysis to study behavior of triple-negative breast cancer cells. She's also, outside of all of this work, the vice president of the CSUSM Biotechnology Organization, and she plans on pursuing a graduate degree in biomedical engineering. So this is all in support of a long-term career goal in pharmacology and drug development. And without further ado, I'm really happy to turn this over to Faheema so she can present on her current work.
Hi. So in honor of October being Breast Cancer Awareness Month, I wanted to share some exciting research that is offering a new light of hope in the fight against breast cancer. So our project is titled Decoding the Tumor Ecosystem, Spatial-omics of Breast Cancer Cells Using Holographic Microscopy. So let's start off by talking about what breast cancer is. So many of us know someone who is affected by breast cancer, but it's also important to understand that not all breast cancers are the same. So when a doctor diagnoses a patient with breast cancer, they are testing for three common proteins or receptors, which are the HER2, estrogen, and progesterone receptors. And now if a cancer tests negative for all three of these receptors, meaning the cells don't have them, it is called triple-negative breast cancer, or TNBC. And TNBC makes up 10 to 15% of all breast cancers. And because they lack these three common receptors, it does not respond to many of the standard hormone therapies or the HER2 targeted drugs, which makes it a more aggressive, harder to treat, and more likely to recur. And that brings us to the core problem that we are addressing. So even with TNBC, there isn't just one type. It is a highly diverse disease. And TNBC has something called intratumoral heterogeneity, meaning that within one tumor, there are multiple different types of cells. And today we mostly classify TNBC using genetic and molecular markers, which are very valuable. However, a static or a 2D view of the genetic code does not tell us or give us the full picture. So our hypothesis is that the physical properties of TNBC cells, such as their shape, size, texture, and their movement, can give us more important information about their behavior and how aggressive they are. So think of it like a personality trait for a cell. But our real challenge is, how are we going to measure these incredibly subtle properties in a living dynamic cell without damaging it? And so this is where our tool comes in, the HoloMonitor live cell imaging system made by PHI Labs. So what the HoloMonitor is a label-free imaging technology that doesn't just give us a flat picture, but a full 3D high-resolution hologram of a living cell. And it does not use any dye, stains, or invasive methods that could alter a cell's natural state. And so how does this work? So as the diagram shows, there's a gentle laser that shines through the cell culture. And when it goes through the cell culture, the light subtly changes, and the microscope captures the small changes in the light and produces what we call the interference pattern. And then the computer compiles all this data to reconstruct and give us a 3D image of the cell. And so the advantage of using the HoloMonitor is that we can watch the cell's behavior over a long period of time, and the cell is also unharmed as this HoloMonitor is in the incubator. So for our project or experiment, we cultured 3 cell lines, which were the T47D, MDA-MB175, and the MDA-MB231 cell lines. And we monitored them for 48 hours using the HoloMonitor. We were able to obtain various parameters from the HoloMonitor as seen on the image in the right. Although we could analyze each parameter individually, we wanted to come up with a way that we could visualize all the key parameters in a single picture. So we wanted to focus on 4 main parameters, which were the area, eccentricity, irregularity, and the optical volume. And this is why we created a k-means clustering algorithm to analyze our quantitative data. So this algorithm is a user interface program, which allows users to interact with the code. So the way the algorithm or the code works is that it will ask the user whether the data is in a single file or in multiple files. So for this demonstration, I have indicated that my data is in a single file. Then it'll read me all the columns in that data file and ask me to identify which column has the cell ID and which columns have the parameters that I would like to study. And once I enter that into the program, it will ask me to identify the number of clusters. So it'll suggest a cluster value and will ask me if I'd like to proceed with that or change the number of clusters. And then once I choose the number of clusters, it'll start analyzing. And so here are the results from the algorithm that we got from the 3 cell lines. And we can see that it classified each cell line as having 4 distinct subtypes. So let's take a closer look at one of the heat maps. So the heat maps are like a fingerprint for each cell cluster. They show the average of the characteristics of the cells within each cell cluster. So the rows are the different cell clusters that the algorithm identified, which are labeled from 0 to 3. And the columns are the specific physical characteristics that we measured for each cell, which were area, eccentricity, irregularity, and optical volume. So area is the size of the cell measured in square micrometers. The eccentricity is the measure of how elongated or circular a cell is. So a value closer to 0 indicates a perfectly circular cell, while a value closer to 1 indicates a very elongated cell. Irregularity is a measure of a shape complexity or how rough or smooth a cell is. So a value closer to 0 would mean that the cell is more smooth, versus a higher value would mean that the cell is more irregular. And the optical volume tells us how dense a cell is, and it is measured in micrometers cubed. So if we look at cluster 0, for example, so the colors are very important when we read this heat map. So red boxes or red values indicate higher values, and then the bluer boxes indicate a lower value. So cluster 0, we can see has higher area, eccentricity, irregularity, and optical volume, which may suggest that they are more dense and they could be potentially more aggressive. And cluster 3, on the other hand, we see has more blue boxes across the board, meaning that they are smaller, less dense, and more regular. And so why does all of this matter? So with this, we could come up with personalized treatment. So a doctor can potentially and precisely classify a patient's TNBC subtype based on the cell's physical traits. So it would allow the doctor to come up with an effective and targeted treatment from day one instead of having to experiment with multiple treatments. And this technology could also offer a faster and more comprehensive way to understand a cell's behavior, reducing the need to do multiple biopsies to better understand the cancer. And it could also potentially aid in drug discovery. So researchers could make medications that target cancer's specific behavior. So the HoloMonitor has allowed us to extract quantitative data and analyze them into separate subgroups to better understand TNBC cells. So the ultimate goal is a future where TNBC patients receive a targeted and personalized and more effective treatment plan, giving them a better chance at fighting this disease. So this project would not have been possible without the incredible support that I received. Huge thank you to my PI, Dr. Luna, for his support and mentorship throughout this whole project. And I'm also very grateful to my lab colleagues for creating such an incredible and great environment. It was great working with all of you all. And special thanks to PHI Labs. Their HoloMonitor technology was incredible to this project. And with that, I'd be happy to take any questions.
And now I want to start by asking if there's anyone from the audience, the Luna Lab, PHI, if you have any questions that you wanted to ask Faheema about any of the slides you presented.
I actually have a questions. And first of all, I would like to say thank you for sharing this amazing work with us. I'm an application specialist from PHI, and I'm very excited to see that our HoloMonitor can help you with that much. And I actually have a questions about the three cell lines that you showed. Are they all triple negative cell lines?
So no, not all of these cell lines are triple negative. So the T47D cell line is an ER positive breast cancer cell line. The MD-MB175 cell line is a HER2 breast cancer cell line, and the MD-MB231 is a triple negative breast cancer cell line. So from this, we can see different cell lines have their own unique fingerprints.
And I have to say, I really like that fingerprint idea you have. And following question is that how should I read about these color clusters that you're showing here? The green, red, purple, how, like, what do they present for?
So let's take MD-MB175 as an example. So in that UMAP plot, which is the plot right above the heat maps, we can see that there are 4 clusters, and we can see that the yellow, green, and the purple clusters are closer together, while the blue cluster is farther apart. So what this tells us is that the yellow, green, and the purple cluster cells share more similarities than the blue cluster, which has more different metrics than the other 3 groups.
Huh. So simply by just looking at the morphology, they are really like behaving very different, and you can really see the different patterns. So they are really, like you said, they are fingerprint. And then I'm thinking like, because they are, it's not only the triple negative cancer cell lines here, but all these can be actually, like, we can do more than just breast cancer. If you do more different cancer cell lines, you probably have different fingerprints that we can use to just figure out which cancer cell line it is. Yeah, I think that is amazing. And then it is fantastic work you do here.
Thank you.
Yeah, it's incredible phenotyping. And also amazing that you're doing this without genetics, right? Because I think that that's how we would normally characterize which is
Right.
everything just based on the morphology here, right? All the data you're collecting here is simply by morphology with HoloMonitor captured.
Yeah.
That is fantastic.
And then I don't know if there's any questions from the Luna Lab. We're so grateful to have you on the call, Carlos. Carlos is Faheema's mentor.
Hi. Yeah, I'll ask Faheema a question. So why don't you tell us again, I would say one of the special characteristics of the HoloMonitor is getting that cell volume that
Right.
you don't really get with a traditional microscope, right? So why don't you
Right.
tell us why is that cell volume telling you about the cell behavior?
Yeah, great question. So volume points out changes in cell behavior, such as the cytoskeleton remodeling, their stress response, and the cell cycle changes. So these are qualities that we wouldn't be specifically able to see with a regular microscope. So yeah, it gives us more information about the cell.
Very cool.
I have to
Thank
say
you.
I'm very sorry.
No, please.
I have to say I'm very happy to hear that as a PHI member, but like I'm very good and also very happy that we can actually help you to find out that data.
Mhm.
So thank
Yeah,
you.
that's great.
And my question is really more about you, Faheema. I'm seeing
Yeah.
so much work in the computational side, combined with the biology. For
Mhm.
you personally, where do you see yourself ending up for your career? Do you want to be more wet lab? Do you want to be more analytical?
I would say I definitely do see myself doing more computational analysis in the future, but I really do love my wet lab as well. But I would definitely say I'm thinking about going to computational biology.
Amazing. I just appreciate this so much. Thank you for sharing your work with us, explaining all the hard work you've done, and yeah, I think there's a lot more to come, I'm sure, and I just appreciate this so much. So thank you. And
Thank
yeah, I
you.
think we can do everyone's on mute, but that's okay. Thank you so much. Okay.
Yeah.